Good. So what I thought today I would give you a slightly different perspective about how we're thinking about functional genomics, and in particular how integrating functional genomics data sets with electronic health records can teach us about essentially disease predisposition and what's the clinical utility. So this is part of a project called Cardinal that was funded by the Chan Zuckerberg Initiative and has partnership with UK cohorts like UK Biobank and Ginseng Health. And so really the point here is to try to think about how we can use functional genomics data sets, but in fact the wealth of new approaches, including, for instance, machine learning, human cell atlasing efforts, and genome sequencing studies to essentially increase the yield of genomic knowledge and the yield of translational insights that we can get from, particularly for common human diseases. And so this prompted this was framed as a pilot project to ask what is the value of embedding single cell RNA sequencing data into cohorts with deep medical record data, and the ability, where is the value of integrating single cell genomic data with all the layers of information, including not just genome sequencing, but also information on demography, clinical phenotypes, health records, and objective disease measurements. And so we, due to scale, but also accessibility, we designed a project that essentially was designed to sample single-cell RNA sequencing from peripheral blood mononuclear cells of nearly 6,500 people from cohorts. And we're interested in this to have a functional genomics readout, but also because, of course, PBMCs, or broadly the immune system, give us insight into age-associated decline in protective immunity and a host of diseases and disease manifestations that can come from that. So potentially the integration of these two types of data can have high translational potential or value, not just essentially in mapping genetic effects, but indeed in delivering markers of, for instance, systemic inflammation or biomarkers of disease. And so I just want to give you a sense of how we're using this data. Of course, there are a million different types of analysis that you can do with this data, and the data will be released to the community for everybody to use. We sampled opportunistically UK Biobank. We targeted 5,000 donors using the MRI imaging recall because we wanted fresh blood. So the individuals at time of sampling had a median age of 67 years of age with a range of 55 to 85. So they are within the stage of life that where we see essentially beginning of immune senescence and decline. And in addition to that, we also sample individuals of Pakistani and Bangladeshi British origin from the Ginseng Health, and these were individuals that were recruited through essentially a bespoke community-based approach. In this case, we recalled individuals on a much broader age range of median range of age of 38 with a span between 18 and 88, so capturing the entire length of the human life. And essentially, the components of that, as I said, is a new single-cell data set, genotype data, GWAS data, and the electronic health records. And I just want to give you a flavor for what we did with that. So starting with cell atlasing, of course, if you want to have single-cell data in thousands of people, you have to have really robust approaches so that you can essentially remove technical variation for experiments that last in the range of a year for data collection, and indeed remove technical variants but preserve biological variants. So we set up, I set up this project when I was still at the Sanger Institute. I've since moved to Italy. But essentially the idea is that we recalled fresh blood from 5 UK sites. Every day the blood was shipped overnight to the Sanger Institute and was processed from fresh the next day. And so in order to do that, we set up a hands-off pipeline that allows to sample thousands of, hundreds of people a day in a very robust way with LIMS control system, etc. And we similarly had a sequencing and then a data processing, at least a quality control processing pipeline that again worked in a fully automated way, so to avoid errors as much as possible. And the resulting atlas, we took a lot of care to make it high quality and clean it, but has nearly 10 million peripheral PBMCs. So these were essentially annotated at 5 different levels of annotation, at 2 levels of cell lineages, and then 3 different levels of cell types and cell states. So CT2 are 15 major cell types. CT2 level, there are 13 main PBMC cell subtypes, if you want. But also we capture 79 distinct cell states that essentially resolve, properties of, for instance, functional activation or immunosenescence. And the data has already been released back to UK Biobank and Genes and Health and will be available to the respective TREs. So if you look at the proportion of cells in the population, we can essentially, so the final data set has nearly 4,500 UK Biobank individuals at 7.3 million cells and 1,200 donors from Genes and Health and 2.5 million cells. And so if you look at the distribution of cells of the population, pretty much the number of cells you see in a person are what you expect given the relative proportion of cells within the sample of blood. And so we have something like 33 common cell states and 29 restricted cell types, so seen only between 1 and 10% of people, and some very rare cell types. So what we did, of course, there's a lot of genetic analysis using a single-cell QTL and rare variant tests. I had actually opted not to give you any of that. I've added a couple of slides at the last minute, but I just want to give you a sense of the power of this data set, but this is not an exhaustive presentation. So essentially, this allows us to go cell type by cell type and map genetic effects in a cell type, as I said, specific format. We've done it on 32 cell type. At the moment, we're repeating analysis now at the cell state level. And essentially to understand which genetic variant affects gene expression in which cellular context. We use it both for biological discovery, that I'm not going to talk about today, but also as a genetic toolbox for drug prioritization, to discover new targets, to understand better, for instance, the mechanism of action, the tissue specificity of expression, and to predict both the target efficacy, but also side effects. And so as part of this work, we've done a very massive discovery data set where we took all the QTLs that we mapped in the cell types, so nearly 500,000 QTLs, and we fine mapped them, and we developed a pipeline for high throughput fine mapping and pleiotropy analysis. We integrated with nearly 20,000 disease phenotypes, and we did a lot of work to harmonize traits and into FO categories to resolve cell type specificity. And then we combine them with drug target information to estimate value of this data for clinical trial support and drug targets. And we compare them with possible with off PBMC effects, such as from GTEx or protein QTLs. And these are just some of the key insights that we get in from this type of discovery effort. We see essentially we have a good understanding of tissue or cell type specificity of genetic effects. We can map how tissue selectivity in QTLs reflect disease context for immune-mediated versus other types of diseases. We can predict a high success rate for drug discovery at different stages of drug development with odds ratio that go between 4.4 and 10 times and 10x. And then we can predict very well drugs with down-regulatory effects, such as inhibitors, antagonists, and blockers, and predict essentially which drugs across 20,000 disease phenotypes will have a high chance of potential adverse events. We hope to release some of the preprints quite quickly, so hopefully this data will become more publicly available soon. So a lesson from here is that essentially this single-cell resolving QTL informed drug target discovery and success in highly specific manners. But I really wanted to focus the majority of the talk on showing how we're thinking about integrating this QTL data with information from electronic health records. And the main question, but there are many subtypes of this, is that can we identify what are the forces or the diseases that shape immune state architecture in our cohorts? So if you take these 79 cell states, you rank them by essentially how frequent a cell is in the atlas, you can begin to look in very coarse ways how whether the cells are more or less abundant in the two cohorts in Genes and Health and UK Biobank. And just want to remind you that the two cohorts differ both by age structure, but also by ethnicity, and of course, also by likely by, for instance, different types of lifestyle exposures or habits. So all of this is currently confounded, if you want. So at broad sense, we see that UK Biobank have an enrichment of central memory T cell states, antigen presentation associated subsets, and CD14, for instance, CD16 monocytes. And these are typical signature of a characteristic of an immune age population with the cumulative inflammatory exposure, so what we would expect to see. In Genes and Health, however, if you see on the left side, we see a higher abundance of innate and naive compartments, which are compatible with the fact that the population is younger, and less memory-skewed immune cells, but with a high prevalence of interferon-activated cell populations. And this is already a first indication that these, for instance, Genes and Health are likely to be younger, but they have potentially a different type of exposure history compared to UK Biobank. And I just want to remind you that these samples were collected post-COVID, so there's of course also a component of that. And so we decided to ask more systematically, what are the population level drivers of these differences? And so if at course level, what we can first do is to derive graphs that describe what is the broad immune composition of our population cohort, and we derive what we call broad archetypes, which are essentially broad definitions of an immune state of a person. And just to take you through some of them, for instance, there's a broad archetype that is defined by an NK CD16 adaptive and other cell types that are typically associated with a higher rate of viral infection. And we can show through a series of analysis that this is indeed potentially a signature of a CMV or other virus infection status, which is predicted, but not measured, but predicted to be more abundant in Genes and Health. There is another signature which is called type 1 interferon state, and by looking into the health records, you can see that the individuals that have the signature add in their health records report of a recent infection, such as a cold or a sore throat. There's also a very strong archetype defined by essentially a CD5 positive B cell subtype that is typical of typically a signature of pre-malignant leukemia that you can see very clearly in UK Biobank, but not in Genes and Health. And other signatures that we can't yet explain, but are essentially not associated with any of these effects. And so what the picture that emerges is that there are dominant immune cell archetypes, broad categories of archetypes that are driven in our data by acute health states that we can map from our data, so for instance, by infection or pre-malignancy state. However, we also know that if you take an essentially immune system, these are, of course, acute events, if you want to, but there is also a more continuous variation in an immune state of a person that is determined by combination of essentially population biology, including, for instance, their age or their demography, long-term exposure, and disease states. And so this kind of picture that emerges is essentially of a continuous gradient of variation, and each person is positioned within essentially this, if you want to, immune architecture space due to the cumulative effect, the baseline characteristics and the cumulative effects of their history. And so we can essentially exploit a large size of this data set to begin to ask more systematically what are the factors that shape the immune architecture as we see it in our data. And I just want to remind you that essentially what we do is we take single cell collection at some point in time in the history of the patients, but for that patient we have already accrued information of demography, sex, age, etc., of exposure, such as whether they are smokers or not, or BMI, and indeed of potential disease events. So what this allows us to do is to model taking essentially the age of the person as a time dimension, differences in immune signatures associated with the healthy or disease states. And so this is the principle. So this is analysis where we look essentially at history readouts of a medical history of a patient. And again, we use a number of approaches to look at this, and I'll just touch on some of them. But essentially, we look at cell compositional signatures, so the absolute abundance of individual cell types in each person. We look at gene-level analysis, including differential gene expression and variant cell composition models. And also, we use machine learning approaches called mixed-mill to try and derive inference that is driven not by canonical annotation, but indeed by the single-cell data as a whole. And so just to give you a flavor of the kind of insights that we see starting with cell composition. So the idea is that, as I said, we model in the life of the population how each of these immune cell types changes. And so we begin to see some very clear effects. So this is a dimorphic pattern in CD14 monocytes, and we see that male donors consistently have higher CD14 monocytes compared to females. We see effects, reproducible effects of BMI and of smoking. These are all very clear signatures. In many cases, they recapitulate what we already know about, for instance, aging of the immune system, but we also, of course, have a lot more resolution than canonical analysis. So we can begin to essentially combine all of these individual signatures into composite signatures that tells us essentially what is the effect of age on immune cell composition in the two populations. And what you see from the overlap of the blue and the green lines here is essentially age, sex, BMI, and smoking have essentially substantially identical signature on shaping immune composition once you account statistically for everything else that is measured. So essentially, the immune signatures for biology, such as aging and sex, and also risk factors, such as smoking and positive, are indeed very consistent across cohorts. However, we also know that these cohorts have very different prevalence of disease. So these are essentially across a number of disease diagnoses. We look at the prevalence in our cohort of diseases in either UK Biobank in blue or in Genes and Health in green. And so, for instance, you see an excess of diseases of metabolism, particularly type 2 diabetes and obesity, and also of skin disorders. And so we ask, okay, now we can account for demography properly. Can we also look at differences that are associated with the carriage of disease state? And I'll ask you to maybe give me a sign when I'm out of time, because I'm not looking. But essentially, we can begin to annotate not just the demography, but indeed the disease programs from the data. And so, for instance, and this allows us to essentially link different diseases to different characteristics of the immune system. For instance, disease characterized by immune reserve loss, immune inflammatory activation, adaptive memory activation, and so on. And so, for instance, what you see from this is simply a similarity plot between the compositional signatures of different diseases, is that there are cases where essentially Genes and Health and UK Biobank have very similar disease signatures, but in other cases they have quite distinctive ones. And so this is just an example. So if you take a cardiometabolic disease, in UK Biobank, the signature associated with cardiometabolic disease are those typical of inflammaging, monocyte CD14 activation, together with B memory and ILC involvement, whereas in Genes and Health, cardiometabolic signals are more similar to a type 2 diabetes and an obesity signature. And this is expected because indeed, for instance, type 2 diabetes in Genes and Health derives from obesity rather than from inflammaging. And similarly, if you take skin inflammation, in UK Biobank is very much a Th17, Th2 expression driven, whereas in Genes and Health, we see signs of cytotoxic suppression. So this is really telling us that although they're essentially risk factors act in similar ways, but then the resulting disease immune maps program depend very much on cohort context, and that includes structure, exposure history, the mix of diagnosis, and potentially other ancestry-linked baseline immunology. We also asked whether this type of program differences can be seen at the level of gene expression programs. And I just want to introduce another, perhaps, complex concept of how we're thinking about that. So if you think about, a disease gene expression biomarker study, you typically look at differences in gene expression which are associated with the disease diagnosis. And so we've seen before with the QTL that in some cases, essentially, you might have a genetic variant that affects gene expression that in turn affects disease, and this is what we use for essentially target discovery. However, there might be also cases where, for instance, the gene expression can be just simply considered a readout of disease state, or indeed there might be other cases where you have a risk factor such as smoking that affects this gene expression and disease separately. And so what we can do now is to take this very rich data set to essentially model statistically exactly these scenarios to tease apart the genetic and non-genetic effects from the data. And so what we do is essentially, first of all, is to try and tease apart genetic and non-genetic effects. For this, we use what we call the variance decomposition analysis, where essentially we model the expression of pseudobulk gene expression as a function of covariates, genetic effects, and EHR history of the patient that is essentially derived from models such as Delphi. In this case, we use one-hot encoding. And then we take essentially, so we remove the genetic effects and we take only the EHR effect. And then we ask, if you take a representation of that EHR-driven gene expression, can we map it back to EHR traits to understand, to map disease programs? And I just want to show you the power of this approach. So this is a plot that essentially shows for each gene, in this case of T cells, how much of the variance in expression of the particular gene is explained by EHR patient histories or by genetics. And so, for instance, if you take this top gene, GPR15, this has 0 genetic variance and completely EHR variance. And what we see is that, indeed, this is a very well-known smoking biomarker. So we essentially deduct through a non-genetic effect, but through the modulation of essentially a CPG site. And so essentially what we can do with this approach is to reconstruct exactly triads of linking a gene expression to an exposure and to disease diagnosis. And in many cases, these triads map to effects that we know from biomarker studies. And so we have now identified thousands of gene exposure disease triads that link essentially exposures to genes and to disease through non-genetic effects. What we can then do is take this non-genetic proportion of genetic gene expression variants and then build essentially gene regulatory programs and ask whether these gene regulatory programs are associated with axes of disease biology and whether those differ between population. And I'll go quickly here because that's essentially a lot of data, but essentially, for instance, this is a case where we identified a program which we call program 1 that essentially recapitulates gene expression variation in the canonical axis of essentially cardiovascular risk progression from BMI to type 2 diabetes to ischemic disease. Another example is a program 2 that essentially pairs atherosclerotic to chronic pain signatures and so on and so forth. So essentially, single cell and EHR inform biological program mapping. And what we can do is to ask whether the two populations deploy these gene regulatory programs in different ways. And this is, we do a kind of a coupling analysis. And this is an example of program 1, this so-called pure metabolic syndrome axis, where we see that essentially both populations have this program, but they use them to different extent. So I just want to conclude, by just pointing to the last analysis. We also use machine learning models that don't require annotation. They go cell by cell and essentially assign the value of each cell in driving or in detecting a disease-related signal. And this is just to show you that the method, again, works really well and allows us to identify population-specific and population-shared disease signatures without prior annotation. These type of approaches are incredibly powerful because they also allow you to partition patients with the same ICD codes in different archetypes that are typically linked to distinct comorbidities. So I'd like to essentially, the kind of insights that we're getting from this type of data is that the EHR-driven integration informs disease program in human population. We show that immune signatures and programs are consistently driven by biology in similar ways and by risk factors. However, the mapped immune programs in diverse diseases depends very much on cohort context. We can dissect the genetic and population consequences at gene, program, and cell composition level. We identify thousands of genes and disease pairs that are linked to both genetic and also non-genetic effects. And we show that these biological programs derive from single cell have population dominance and differentiate patients with different comorbidity profiles. And I just want to say, I haven't shown this, but also EQTLs, when we incorporate information from health records, we can identify context-specific EQTLs similarly to what described before. And so essentially, this is, I think, a powerful, if you want, representation of how single cell applied to population level can drive understanding of genetic and non-genetic factors driving chronic disease risk in populations. We created a high-resolution, high-quality data set that will be released to the community, and we very much hope that this will be used a lot. And so with this, I'd like to thank all my collaborators. I haven't been able to nominate them, but the names were on slides. And thank to you, and thanks to our cohorts and funders, and thank to you for your attention.